Okay, welcome to our first online lecture for BIO367 Microbial Diversity. And this is going to be lecture 3, and it is the second part of a first-part lecture series on bacterial and archaeal cell structure. This will be part A of this lecture. So the class goals overall are going to be to appreciate the functions of the cytoplasmic membrane, particularly within the context of prokaryotic organisms, those belonging to the domains Archaea and Bacteria, to know how the cytoplasmic membrane differs between Bacteria and Archaea, to appreciate the variety of cellular inclusions used by Bacteria and Archaea with regards to their structure and function, and to understand the structure and function of endospores. In this A part of the lecture, we'll cover objectives 1 and 2. So the cytoplasmic membrane is essential to all cells, and you should be very familiar with its ability to act as a selectively permeable barrier. So that's the function shown there on the left. That's a barrier that's incredibly important in all cells on Earth, and I really don't need to elaborate on it here. Also common to all cells is the fact that cytoplasmic membranes are about half lipid, which means the phospholipids found in the membrane, and about half protein. So we have a lot of proteins involved in transport, bioenergetics, and chemotaxis found in cytoplasmic membranes. Now the function that I want to particularly have you focus on is function C, that of energy conservation. Okay, so if you think about the fact, the electron transport chain and what you know about that, you should remember a little bit about aerobic respiration from your earlier biology classes, and you should remember how electron transport chains serve in the process of aerobic respiration, and you should be familiar with the term proton motive force. I'm not going to review all of that here, but I want to point out to you that in eukaryotic cells, that electron transport chain is found in the mitochondria, which are organelles within the cell. However, in a bacterial or an archaeal cell, you have a proton motive force established at the cytoplasmic membrane. So we have a proton motive force. We have a set of proteins and organic molecules that are the electron transport chain. It's maintaining the proton motive force, and therefore in the cytoplasmic membrane we have an essential protein called ATP synthase. Okay. In bacteria and archaea, that is not found in the inner mitochondrial membranes because they don't have one. It is instead found at the cytoplasmic membrane. So energy conservation, which means to generate and use a proton motive force in order to make ATP, that happens at the cytoplasmic membrane in bacteria and archaea. That's a really important thing to remember as you go through learning about microbiology. All right, so what are these membranes like? You're familiar with the structure of a phospholipid, so this should be review for you. We have a fatty acid tail. Okay, remember that at each of these junctures we have carbons, and if those are single covalent bonds connecting those carbons together, then you could count off the hydrogens that would be on either side. So for example, we know here we've got a hydrogen, it's supposed to be a hydrogen, and here on the other side we've got a hydrogen. It's a bit messy, and then the other two bonding requirements for carbon are met by single covalent bonds with the neighboring carbons. That's a very hydrophobic structure. It is connected through ether linkages found in here to a glycerol structure. The glycerol structure is shown in blue, and that is also including something called ethanolamine, shown in green, right here. Very common structure seen in phospholipids for bacteria, very similar to what we see in eukaryotic cells. So bacterial, phospholipids, are like eukaryotic phospholipids. And they're going to organize themselves in the characteristic lipid bilayer that you see in the B part of the figure here, okay? Aligned, so they're hydrophilic, phosphate-containing heads, shown in blue, okay, are interacting with the watery environment inside and outside of the cell, or perhaps at the periplasmic space, and the hydrophobic fatty acid tails are hidden from the water. So that should just be kind of business as usual for all of you, and not really serve sort of more in the context of review. All right, so another thing to remember is that bacteria do not have cholesterol. So cholesterol is something that is found in eukaryotes. Instead, bacteria use other structures. One of the common ones that we know about are the hopanoids. So bacteria, many of them make structures called hopanoids. These are very planar or flat molecules. They contain a lot of ring-like structure. There's a hopanoid molecule here, and they serve the function of stiffening and decreasing the permeability of the membrane. You might want to do that in circumstances where you have an organism that has adapted to live at very high temperatures that would otherwise cause the membrane to basically dissolve. If you include some extra hopanoids there, then you will have a stiffer, less permeable membrane, and the cell will better be able to control what goes in and what goes out of the cell. Okay, let's think a little bit about the phospholipids of Archaea and how they differ from those found in bacteria and eukarya. So over on the left, over here, we have, sorry about that, a phospholipid from bacteria and eukarya. These R groups signify the point of attachment for the hydrocarbon tails, okay? And what you'll notice is that, and over here we've got the 3 carbons derived from glycerol. So the key here, the thing that I want to note, is shown in pink. It's this ester bond. That's a very characteristic kind of covalent bond that is associated with the structure of phospholipids as found in bacteria and eukarya. And the thing is that in Archaea we see something different. In Archaea we have similar phospholipids, so we have the phosphate group similar to over here. We have a glycerol type backbone, and we still have hydrocarbon tails. Although note that the hydrocarbon tails are made out of isoprene units, as shown here for C. That's a slight difference between phospholipids in these organisms. And the other important difference is that those hydrocarbon tails are attached to the glycerol backbone not by ester linkages but by ether linkages. And that might seem like your microbiology professor making much ado about nothing, but if you think about the fact that forming all those chemical bonds shown requires catalysis that is assisted by enzymes. And each of those enzymes is encoded for by 1 or more genes. The fact that we have ester bonds in bacteria and eukarya, and instead we have ether bonds in Archaea to do this basically the same job, shows that we have fundamentally different enzymes and therefore different genes, and it's an indication of the different evolutionary histories of these organisms, the Archaea, as compared to the bacteria and the eukarya. Remember, the domain Archaea is quite evolutionarily distinct from the other 2 domains. As we continue looking at the archaeal membrane structure shown in this slide, we see some other really interesting differences. And what I want to point out right now is that many of the archaea on Earth are found in extremely harsh or, extreme environments. So for example, many of them are found in very high temperature environments. So organisms that can survive temperatures at or above boiling, 100 degrees Celsius. When you find organisms in that category, they are typically archaea. So that's quite interesting. And they have adapted to be able to survive those high temperatures. They have particular kinds of enzymes with adaptation, and they also have adapted so that their phospholipid-based membranes are different. And one of the structures that we see is a structure referred to as a unit membrane. Okay, this is not a bilayer. It's a unit membrane, unit being one thing. And if we look here at this structure and this structure, you're looking at the kinds of phospholipids that make up a unit membrane. So rather than having separation at this point so that you have distinct phospholipids here and here, instead they are connected all the way through. Okay, and they're going to be oriented into a membrane-like arrangement, but they're going to be much more rigid because of that connectivity. And that rigidity, that is associated with a unit membrane, allows them to resist becoming overly permeable and entirely dissolving when confronted with extremely high temperatures. Similarly, some of the ring-type structures found in the Archaea are able to make the membrane more rigid. So all of these adaptations increase rigidity, and they help under high temperature conditions. Shown down here is a picture of the lipid monolayer or the unit membrane as compared with what is more typically observed over on the left in organisms that live at what we think of as more normal temperatures. So these are some really interesting evolutionary adaptations that we've discovered through studying organisms from very hot places, volcanic sediments, and heated areas within the context of deep-sea hydrothermal vents. And even some human environments where high temperatures under certain industrial conditions have selected for interesting archaea. With that, I'll close this part of the lecture and leave you to start part B.